Connecting to cloud is recording.
Okay. So I see that the participants are coming in, attendees.
Yes, it's wonderful. Welcome everybody. We will start soon. Wow, I think this is going to be exciting.
Wow.
Yeah,
the number of attendees is going
it's
up.
like going, yeah.
That's fantastic. Wow, we have a big crowd, eh?
Yeah. Okay, we'll probably wait a couple of minutes as people are signing in. It's 11:59. Okay. Elena, it's 12 in New Haven, in New York, in many places.
Okay, it's 9 a.m. in Pacific time here. So, okay, so I think that we can start, and we see that more people are coming in, and that's fantastic. I am Elena Zúñiga from the University of California, San Diego, and I'm super excited to be here today virtually with my dear friend and co-organizer of the Global IMMUNOTALKS 2020, Carla Rodlin from Yale University, and our amazing speaker today, Daniel Mucida from Rockefeller University. And so Carla and I would like to welcome every one of you today to this new seminar series. We feel very excited and also very honored to be launching this seminar series. And we thought that it would be important to share up front which are the goals that we had in mind and we would like to accomplish with these talks. And so the first goal is to benefit and inspire immunologists across the world in an egalitarian manner, or as egalitarian as possible. And so for that, we have committed to make these talks accessible for free. So no, regardless of the financial means, everyone can have access to excellence in immunology, and that will be presented by the speakers. In addition, we will record the talks and we upload them in the Global IMMUNOTALK webpage, and also in a new YouTube channel named Global IMMUNOTALKs. And this will allow people with scheduling conflicts because they live in a different time zone that is not a good fit. In Australia is 2 a.m. Or people that have a scheduling conflict due to family obligations or due to work obligations, they can still benefit from these talks. And last, to make sure that people that watch the talks live or recorded, they can participate equally to the discussions. We are making the discussions and the questions and answers via Twitter, the new hashtag Global Immuno. And these discussions are expected to be among attendees and also contingent on speaker availability, also with the speaker. And so we hope that making the talks free, recorded, and making the discussions virtual via Twitter, we will be able to benefit with these talks, or our hope is to benefit with these talks a large number of students and scientists across the world, regardless of their financial means, of their time zone, family obligations, work obligations, etc. And so a second goal, equally important, is that we are hoping to, with these talks, increase opportunities for scientific learning without traveling. And so these will allow accommodating people with travel restrictions, which unfortunately, due to the COVID-19 pandemic, this is all of us now. When this pandemic is over, and we certainly all hope this is very soon, when this pandemic is over, there will still be people that have travel restrictions because of financial limitations. It's expensive to travel because of family obligations, work obligations, that they find talks beneficial. In addition, and very important as well, is that by providing opportunities for learning without traveling, we are hopefully contributing to tackle climate change, and by diminish carbon emissions, and making an opportunity for learning in an environmentally friendly manner. So none of these would be possible without the greatness and generosity of scientists in our community that have agreed to participate in this program and spend their time preparing the talks and sharing their research. And so we are deeply thankful to all the speakers that have agreed, all of the ones listed here, and more speakers that have agreed, and we are studying for the months after August. So thank you so much for your support. Carla and I fully recognize that all the credit should go to speakers for their generosity and participation. So in addition to thanking the speakers, we would like to thank Philip White, who started a similar seminar series on metabolic physiology that has been going on since March 17 and will end, I think, at the end of April. These are daily talks, and he was an inspiration for us, and he provided advice. We want to thank Philip and recommend you to go and watch the talks. They are all recorded, and with the hashtag #FIS2020. So thank you, Philip, for your coaching and help. So last, some practical notes. All the communications for this seminar series will be done via Twitter at the hashtag Global Immuno from my account. And we will also post them in the Global IMMUNOTALK webpage. Again, this webpage is in the URL at the bottom of the post that many of you received. And so We hope everything goes as we plan, but we ask you to please be patient if we make any mistake or if we have any unforeseen technical issues. We will try to solve that as soon as possible and communicate with you again via Twitter or announcing in the webpage and try to solve that soon. So that's all what I have to say, and with this I will pass the virtual microphone to Carla so she can continue.
Thank you, Elena. Thank you so much. This is so exciting and so humbling. We have more than 1,800 of you joining us from everywhere in the world. So it's very, very special. I would first like to thank Elena. She's one of my dearest friends. And when she contacted me about this idea, I could, of course, not say yes to it, right? It's a phenomenal idea. Now, it is my real pleasure to introduce our first speaker of this series, Dr. Daniel Mucida. Daniel is the head of the Laboratory of Mucosal Immunology at Rockefeller University. But to me, he's Danny, and he's also a very dear friend. We met a long time ago in San Diego, and he really introduced me to mucosal immunology. Now, Danny started his career in Brazil and continued his stellar career here in the United States. As you will see from his work, he always pushes the boundaries, and this is really something that I admire about his approach to research. His work is also very elegant, and I think that all of you are in for a treat today. Now, Danny is also very socially committed and is not afraid to speak his thoughts. So it's something that I very much appreciate as a colleague. So Danny, it's a real pleasure to have you join us. Thank you for being our first speaker in this series.
Okay, thank you, Carla. I'm going to share the screen. Let me know if it's fine.
Yes, that's great. Now, before you start, Danny, as you know, we have many young trainees joining us from everywhere in the world. So before we start with your scientific talk, I'd like to ask you if you could go back in time and remember the 20-year-old Danny. What could you tell to yourself after the experience that you now have?
When you guys initially mentioned this question, I actually didn't think about the scientific tips. I think people should always follow, interesting questions in biology, regardless on whether it's a hot area in the topic or regardless whether it's currently fun. But when you guys thought about this question, I was actually thinking about the situation we are in the world today, and I would point two moments that influenced a lot. One was Bush versus Gore, where presumably it was sort of a tie in Florida, and then Bush, via the Supreme Court decision, managed to get the edge and be elected president, and we know what happened after that. So that, I think, was a turning point. Was just after I turned 20, and that kind of came to my mind. And if I could go back in time, I would definitely try to spread the fact that this guy could be elected, and a lot of bad things would happen, and it's just a few thousand people maybe would make a difference. And the second, I would say, is what happened to the Trump election, 2016, that also most of us didn't want to believe that this could happen. And the same thing is actually happening in England with the Brexit, as well as in Brazil with Bolsonaro being elected. And I couldn't stop thinking that, if more people were aware that, extreme right and fascists could rise at that level, I couldn't stop thinking that I think would be the tip I would give to myself, is to make people aware that this can happen. Is a very serious threat, not only for science, but also for the environment, as you know, you guys discussed about carbon footprint. So anyway, I'm sorry it was not a scientific tip I gave, but that's what came to my mind.
Thank you, Danny. It's always nice to hear your thoughts. We look forward to your talk. Thank you. Thanks for being our first speaker.
All right. So thank you, Elina and Carla. I also congratulate you guys for organizing this. This is something that actually there were discussions before whether this should be, you know, more common in science, that people avoid a lot of travel, particularly in large conferences like, you know, ACR or AI, that, you know, 20,000 people travel to a particular place. I think we definitely can discuss and rethink whether all this travel is necessary. I'm sure scientific talks are important and meeting people is important, but we can try to reduce in a level that is, manageable for the environment as well in terms of carbon footprint. So anyway, I will start the presentation after thanking you guys. And just to point out that I'm going to give a summary of some recent work that we've been doing in the lab on neuro-immune interactions. But I like to always start with, particularly because they are trainees in immunology, listen to this presentation. And I like to make analogy with the immune system in a political situation. And the point here, which is raised by many groups, and I think, Rosa Medjitov and Miguel Soares in Portugal have really brought this to mainstream immunology, which is, in addition to resistance against damage of the pathogens, you know, we also need to develop a tolerance against our own immune response that is triggered upon an encounter with a pathogen. And this process called disease tolerance is analogous to what we can bring to the political situation today, where we see foreigners as invaders, potentially dangerous invaders. And in many cases, our reaction to that, it's more serious than the invader itself. And this is also actually the case of the coronavirus crisis here, where it's thought that not the virus itself, but rather the immune response triggered to the virus is what is causing most of the pathology observed in COVID-19 patients. But, you know, the lab, my lab and any other lab studying in mucosal immunology face actually a different situation, which is the fact that the vast majority of non-self interactions, and because the mucosal immune system, you know, comprise the largest surface of the body, and that is the case then in the whole body, is we are encountering non-self molecules or organisms that are not harmful. They are mostly harmless or beneficial to our body. So against those, rather than develop resistance, we need to develop tolerance. And I think that can also be translated to immigrants coming to our countries, that, you know, the really vast majority of them are beneficial to the host or to the country. So with that analogy, I'd like to mention that this is the question that lab studies, how to achieve tolerance and protection without jeopardizing the integrity of the tissue. And you can see here several topics that my lab is studying, from the epithelial layer of the intestine, where we study the adaptation of these immune cells that migrate to this layer called intraepithelial lymphocytes, and the function of these cells and how can they adapt to this environment, to also systemic effects of luminal stimulation, which can be seen as a process of oral tolerance, which is depicted here by the mesenteric lymph nodes. But today I will, as the title of the talk says, I will discuss a third topic of the lab, which is to define neuroimmune interactions in the intestinal environment. The gut, as you see by this presentation, a large amount of neurons, in addition to this high concentration of immune cells. So it's an interesting place to study neuroimmune interactions. And what I'm going to present today, it's a work that was mostly developed by Paul Mueller and Fanny Mattis. It started before with another student from the lab, Ilana Gabani, but I will focus on these two studies, one that was published rather recently, Fanny and Paul Koffer's author, and I will then present a study that is in press, but is already available in bioarchives, discussing a follow-up of characterization of sympathetic innervation. These are some of the people that helped us along the way. and our lab is really thankful for generosity of these groups that shared mouse strains, insights, as well as tools really to characterize these neuroimmune interactions. Zach and Tomasz also mentioned here are people that have helped these more recent studies and continue to explore, potential questions that these initial observations raised. So just to divide the talk in two parts. The first would be the functional consequences of neuroimmune interactions, particularly macrophage and enteric neuron interactions in regards to enteric infections. That's the first part of the presentation. And then I will describe our recent studies describing how the gut sympathetic innervation can be modulated by microbes in the intestine. So I like to start with the anatomy, describing where the macrophages and immune cells are present in the gut, which is mostly in the mucosal region. And you can see here the lamina propria macrophages in red here, really occupying dense regions of the intestine. And you can see there's a separation around the submucosal region, and then you have this muscularis macrophages. The important point here is that these macrophages in the lamina propria are really nearby the luminal stimulation microbiota as well as dietary antigens. And you have these ones in the muscularis, which are far from the luminal stimulation, but they are nearby neuronal cell bodies. If you look at this animation that Paul did using also techniques, and you can see here that we are focusing particularly in the mucosal region of the intestine. You can see in green are macrophages. In purple or bluish, you see the epithelial lining of the intestine, single layer of epithelial cells, and you're going to see as the movie progresses, neuronal processes. You see the dense population of macrophages, how the neuronal process extend all the way to the tip of the villi, and then you have down in the muscularis region, the neuronal cell bodies, which you can see here. So the initial observation that we did made several years ago, which came after the description of these functional neuroimmune interactions by Miriam Erad and Elena Bogunovic, that basically you have this macrophage sitting on top of neuronal cell bodies, and there appears to be several potential functional consequences of this proximity between these cell types. And what we showed initially, and that's just a summary of our work that was published several years ago, is that these macrophages located in the muscularis region of the gut, they are at steady state already expressing a program that is tissue protective program, what people call M2. It's not a typical M2, but it's the gene profile resembles M2 profile. And this can be enhanced or reinforced upon enteric infections, where macrophages, upon sensing of norepinephrine via beta-2-adrenergic receptors, can further upregulate this M2 profile or this tissue protective gene profile that they express. So, of course, the question that we raised after this observation is exactly what are the functional consequences of this gene profile that these macrophages express nearby neurons, and whether this may be playing a role in the protection of neurons during inflammatory triggers. So what we did was to come back to this initial slide where we are studying the balance between resistance and tolerance in the gut. And in terms of examples or clinical examples of where this can be relevant, it's really one of the main ones would be the so-called post-infectious irritable bowel syndrome, which it's accounted roughly to about 10% of enteric infections that will develop the symptoms, which include visceral pain, altered fecal output, which incorporates both diarrhea, which is accelerated peristalsis, or constipation, as well as neuropathy, damage in some level of the neurons located in the intestine. And several pathogens have been shown to trigger this post-infectious IBS in humans, including Shigella, Salmonella, and Campylobacter. And the important feature here is that these infections primarily get cleared either by antibiotic or by host resistance mechanism in case of humans. However, the symptoms appear in general after the pathogen is cleared, where you have this residual inflammation. So what we decided to do in the lab was to check several enteric pathogens, whether we could mimic some of these features in a mouse model, so we could study mechanisms of these potential interactions between neurons and immune cells. So that's the summary, and I'm going a little bit fast in this first part of the presentation because it's, as I said, was published a couple of months ago. But basically this summarizes why we chose this model, which is using an infection of a mutant Salmonella strain called SPIB, which is able to invade the cells, but is unable to proliferate inside the cell. So that makes this a non-pathological, a less pathogenic model of Salmonella infection, because the Salmonella, as you can see by the left, in the left graph here, it's cleared in about a week after initial oral inoculation. At the same time that the Salmonella is being cleared, you see a peak in lipocalin-2 levels in the feces, which is an indirect measure of how the inflammation is being triggered in the tissue. So you have a peak of inflammation around the same time that the Salmonella is being cleared. That is a typical resistance mechanism that is being triggered in the host. However, what we noticed, and that is an important point for this model, is that even though the Salmonella was cleared in about a week, we see a residual inflammation, again, as measured by the lipocalin-2 in the feces, that persists for weeks after the initial clearance of this bug. And an indirect measure of whether this could be affecting additional intestinal functions, we had by measuring the total transit time, which is the time that takes for a dye containing in a diet to come from feeding until it comes out in the feces. So that's called the total transit time. We did not see any change in transit time or significant change in transit time early during the infection course, so about a week. However, after the pathogen was cleared, and that you can see the time point, the 12, 17, 70 days after infection, we see a persistence increase and delay in the transit time, suggesting a symptom that is resembling, again, post-infectious IBS in the constipation module of this syndrome. So that shows a bug that is less pathogenic than wild-type Salmonella, that leaves residual inflammation and also is associated with indirect measures of neuronal activity. Of course, it could be also muscular activity as well. So for people that are not familiar with the anatomy of the intestine, in both humans and mice, you have the proximal small intestine connected to the stomach. You have the duodenum, which is low in terms of microbial diversity and number. However, is where most of the food is absorbed. And then you have jejunum and ileum towards the distal small intestine. And then you have large intestine, where you have a spike in terms of number of microbiota species, as well diversity in the large intestine, which is about 1,000-fold more than what you have in the small intestine. And in this region, in the connection between the ileum and the cecum, is where most of the Salmonella is able to invade and cause inflammation. So what we did was to measure and count the number of neurons as a direct measure of a potential neuropathy or damage of enteric neurons that would occur after this enteric pathogen infection. And what we observed was that if you measure the number of neurons in the duodenum, in the proximal small intestine, again, where the Salmonella is not known to cause damage, is that you have a similar number of neurons in the myenteric plexus of the duodenum, which is the main plexus, neuronal plexus of neurons located inside the intestine, like in the cell bodies located inside the intestine. However, if you measure this same neuronal numbers in the distal small intestine ileum or in the colon, you see a substantial loss of intrinsic enteric neurons, about 25 to 30% loss, very quickly after the initial infection. And this persists for many weeks after the bacteria is cleared. So you can see here one example of a measure of 130 days after the initial infection, and we still observe this decline in the number of enteric neurons in these animals. I'm going to come back to this topic later in this first part of the presentation to whether you have any way that you can regenerate or recover these neurons. In this case here, without manipulation, we saw a persistence in the loss of enteric neurons. Okay, so we have
Danny,
at this point.
Danny, this is Carla.
I think yes.
Can you hear me?
Yes.
You have in the lower right a box. Do you see it? It says no build effects. Do you see?
I don't see it.
Okay, no. Okay, sometimes it covers some of your data. Okay,
I didn't see that. Okay.
Let's see if now we can move forward without it.
Okay.
Now I don't see it.
I don't know. Yeah.
See? Now it's good. Thank you.
Okay. I don't know if it was part of the keynote.
We could see most of it, don't worry. But now it's good.
Okay, so we asked then whether how these neurons would be dying, because it's a fast cell death mechanisms, and initially we hypothesized would be via caspase-3. We did not find evidence of caspase-3 mediated cell death. So what we decided to do is use a tool that we have been using in the lab for several years. And most of neuroscientists have been using this for many, many years, which is called translating ribosomal feed purification, or TRAP, which is based on a ribosomal protein that is tagged, that you can use to investigate the transcriptomics of cells, and in this case, it's going to be neuronal cells in the cell sort independent manner. So basically, you have the tissue that you can isolate, and you have these tagged ribosomes with, in this case, with HA, that you can come with anti-HA beads and isolate the ribosomes and perform transcriptomic analysis. So of course, the advantage of this over cell sorting is that you don't expose neurons to this disruption of the tissue. Of course, many people have been using single nuclei RNA-seq. We opted for this active translating mRNA profiling. We found that this it's very efficient for both intrinsic neurons when you cross this ribotag, which is a sort of a trap approach, with a SNAP25 Cree mice. So therefore, this is a general neuronal driver. And then we can observe the expression of HA, which is what is tagged the ribosome, both in intrinsic neurons located inside the intestine, as well as the extrinsic sensory neurons, as an example here. I'm going to come back to that in the second part of the presentation. But we use this tool to really look whether we can find machinery that could be engaged upon infection that would lead to neuronal loss. And what we found, and I'm not going to explain details, all the confirmations for that, just suffice to say that Richard Flavel's group at Yale have also observed a similar machinery, which is caspase-11, and likely upstream of that, NLRP6 of the inflammasome pathway, that presumably would lead both or either to pyroptosis-mediated cell death or to the production of inflammatory cytokines, IL-1, IL-18, and we found the expression of this using exactly this trap approach that we have been using in the lab for several years. And I just like to show two confirmations that we used cell intrinsic tools to confirm that neurons would be engaged in this pathway after enteric infections. One of them it's using AAV approach, which we inject this AAV in circulation, is tropic for peripheral neurons. So you infect most of peripheral neurons. And what we did was, in that time, we did not have the conditional knockout of caspase-11 or caspase-4. So we injected AAV expressing Cree into the circulation. And then this AAV would infect neurons and excise and flux caspase-11 specifically in peripheral neurons. That you can see here, because this AAV also expressed a GFP that is indeed targeting, and we calculated about 80% of enteric neurons are targeted by this approach. Therefore, we excise and eliminate caspase-11 in a cell-specific manner. So when we infected animals that were exposed to this approach, and you can see here both the neuronal count in the ileum and in the colon, we found that the expression of the Cree virus that would excise caspase-11 would prevent infection-induced neuronal cell death, again, reinforcing the possibility that these neurons die via this likely inflammasome pathway, which is engaging activation of caspase-11. We have also used a conditional knockout approach then to address whether the upstream mediator would be NLRP6, which is a non-convention, or at least less conventional than NLRP3. And indeed, if you flux NLRP6, again using a SNAP25 Cree, which is a general neuronal driver, we saw that if we infect animals that are Cree positive and flux NLRP6, there is virtually no loss of enteric neurons post-infection, while the control mice Cree negative or flux plus show the usual at about 20 to 30% loss of enteric neurons. So that suggests that this is being engaged post-enteric infections. So just to come back to the initial observation I mentioned initially of this presentation, that the neurons also can be potentially protected by a mechanism involving beta-2-adrenergic signaling in the muscularis macrophages. What we did, this is just an example of the innovation that's coming from outside testing of sympathetic neurons. That's was also published in our initial study 4 years ago. And just to show that these TH fibers tyrosine hydroxylase expressing neurons are primarily concentrated indeed in the muscularis region where these macrophages are located. So we did several approaches to address whether this communication would play a role in this neuronal cell death post-infection. And you can see here that upon infection with SPIB, if you treat animals with a ligand of adrenergic receptor, an agonist of adrenergic receptors, beta-2-adrenergic receptor, salbutamol, during the course of infection, you completely prevent neuronal cell death after SPIB exposure. And we did an additional control for that using a tool that was developed also by Miriam Merad's group using anti-CSF receptor antibodies. And if you do that and provide the same ligand, so if you deplete the macrophages, and this is a preferential depletion of muscularis macrophages, if you titrate these antibodies, therefore it's the macrophages in the muscle were mostly absent. And if you provide the ligand, salbutamol no longer was able to prevent neuronal cell death post-infection. So that shows that it's very likely that norepinephrine is engaged in this circuit, that norepinephrine can signal very likely to macrophages, and macrophages engage a tissue protective program that prevents neuronal cell loss. To confirm that, we crossed myeloid driver lysozyme M3 with adrenergic receptor flux animals. You can see here that the clearance of the salmonella is maintained in the control and the flux animals. However, when we count the number of neurons post-infection, you see that in steady state here on this left side, there is no change in the number of neurons in the control and the flux animals. However, if we infect these animals, you will see again an accentuated or aggravated cell loss of these enteric neurons post-salmonella infection, again suggesting that the macrophages are the ones that need to be sensing norepinephrine, and the sensing of norepinephrine would trigger some protective program in the macrophage that would allow for the protection of the neurons. Of course, if you do the control, providing the ligand of norepinephrine to these animals, what we see is that indeed in the control mice, you can prevent neuronal cell loss. However, in the mice lacking adrenergic receptors in the myeloid compartment, there was still the accentuated or aggravated cell loss upon infection. Okay, so the final part of this project was try to investigate how the macrophages would be engaging these neuroprotective mechanisms, and this, we suspected very much on the path of polyamines. First, because the group of Flavel and others have shown that likely polyamines are upstream in preventing an RP6 mediated inflammasome activation, and additionally because polyamines themselves are known to be neuroprotective. So there are potentially two non-exclusive pathways that could be engaged in polyamines in helping neurons not die during infection. And we also had evidence that the macrophage would upregulate arginase 1, which can engage, which can be used for the polyamine production in these macrophages. And this was, again, as you can see by this data, when we use mice lacking adrenergic receptor, this was dependent on signaling of norepinephrine in these macrophages. So what we did was to first use a pharmacological approach to inhibit polyamine secretion using the FMO, which targets ODC1 mediated polyamine secretion. And if in fact the animals that were treated with the FMO, we see again an accentuated loss. Therefore, if you prevent polyamine secretion, there's more loss of enteric neurons. If you provide one of the main polyamines available in the body, spermine, to these animals during the course of infection, there is a partial, it's not a total loss, rescue, but it's a partial rescue in the number of neurons that are dying. And finally, if you cross an arginase 1 flux with a myeloid driver, you have a very similar neuronal loss that you see in animals lacking the adrenergic receptor, again suggesting that arginase 1 upstream of polyamines would be engaging as a pathway that from the signal of norepinephrine in these macrophages. So a summary of these observations here is that the initial finding that these macrophages located nearby enteric neurons may play a functional role in preventing this because they express high levels of tissue protective program. And then we investigated again in the models of infection that we observed several different pathogens are able to trigger neuronal cell loss in the gut. And we found that engagement of this pathway and dependent on the sensing of norepinephrine trigger this protective program in the macrophage, which depends on polyamines that then work in preventing this vast neuronal cell loss. Okay, so there are two outstanding questions in this first part that I like to present on mostly unpublished data. And these are two important questions that always came out in talks that I gave in the past few years. And Paul and Fanny have really made a very nice effort in addressing this question. And the first one is regarding, how come that we don't see any recovery? It is thought to be that enteric neurons are able to recover doing damage. And we kind of were puzzled about that. And we came back to, we kind of went to this literature where there are described loss or reduction in the numbers of enteric neurons in animals lacking microbiota. And we could confirm that by providing broad spectrum antibiotic to wild type mice. That's a very fast and accentuated loss. It's a bit less severe than what we have in Salmonella, but it's about 20% loss of the number of enteric neurons by providing broad spectrum antibiotics. If you remove the antibiotics and let the microbiota recover, or if you colonize with a pre-infection microbiota composition, then we could see a recovery in the number of neurons, suggesting that the composition of the gut microbiota may be modulating the number of enteric neurons that you have in the gut. So to address whether this was the case also during infection, what we did was to compare animals that were infected with SPI-B and then not treated versus the animals infected with SPI-B that we replaced the post-infection microbiota with a pre-infection microbiota. We treated these animals with vancomycin, ampicillin, metronidazole, neomycin, so a combination of broad-spectrum antibiotics, and then we colonize these animals with a pre-infection microbiota. And by doing that, we are able to observe a rescue in the number of neurons post-infection, suggesting that this persistent loss that we see in enteric neurons can be actually triggered and modulated by the microbiota. It's something that we are now trying to investigate how the mechanisms of that and whether a specific species of bacteria are involved. The second question also it's quite relevant, and it's quite relevant for the moment we are living, and a little bit deals with this picture here. So this picture was taken in a Mar-a-Lago party that the Brazilian delegation, you can see here, Bolsonaro, the Brazilian president, and Donald Trump. In the beginning, this was in March 7. If you guys remember, US had already cases of coronavirus infections. Brazil had very little, I would say in the dozens or less cases. And this guy in the back here actually was in the party, all these guys in the party, and he was tested positive. By then, Donald Trump was still downplaying the relevance of this infection. Bolsonaro, believe it or not, is still downplaying the relevance of this epidemics, of this pandemic. What was perhaps pretty sad to Brazil is that this delegation and this reckless party that was taking place in the middle of a pandemic led to about 22 people in the Brazilian delegation that tested positive after this party. And in that time, Brazil basically had a few dozen cases, which suggests that this party between the fascist governments of Brazil and US triggered a spike in the number of cases in Brazil. So the problem here is that it suggests that both Bolsonaro and Trump have a neuronal problem. And the question that the lab wanted to ask about this is how low can the number of neurons go? And that's called the Donald Trump Bolsonaro test. If you keep infecting animals, can they keep losing neurons? So one day they reach the number of neurons that these guys have in the brain. And to test that, what we did was to compare two different pathogens that we know cause neuronal loss. Yersinia pseudotuberculosis, that was used by Yasmin Belkade's group and Igor Brodsky in a really remarkable paper showing immunological scarring. Yersinia, as you can see here, triggers about 15-20% loss of enteric neurons in the same region as the SPI-B, which triggers a bit more severe loss. And you can see here, however, if you infect animals with Yersinia and come with SPI-B, there is no further loss of enteric neurons, suggesting that something happens in the tissue that prevents this continual loss on consecutive infections, which is something that my lab is also trying to investigate the mechanisms of that. One indication of what are the mechanisms, Tomasz Arends, who is a postdoc who is studying this project now, had was that when he did the same experiment, but deleted using anti-CSF1 antibodies, the muscularis macrophages, if he does that, then he sees that this consecutive infection would continue to trigger loss of enteric neurons, as you can see here by this comparison. So if we infect with Yersinia, delete the macrophages that we believe are important in controlling neuronal loss, then the mice, upon SPI-B infection, have an additional loss of neurons, suggesting that this tissue preparation during consecutive infections is likely mediated by the muscularis macrophages as well. Of course, we don't know exactly what is being imprinted in this macrophage that persists for a long time, but that's something that we are trying to investigate. Okay, I will go to the second part of the presentation, which is, you don't need to worry, it's not going to be as long as the first part, but it's basically a more neuronal side of the talk, which deals with the fact that if you paid attention in the first part, that there's something happening in the extrinsic innervation of the intestine that is releasing norepinephrine in the gut. And the general question we ask is, how can microbes that are located inside the intestine or pathogens that are infecting the intestine can regulate the gut extrinsic sympathetic innervation, which provides norepinephrine signaling to the intestine? This is another image of the intestine. And you can see here, this is another iDisco image of the ileum. And you can see this standing for the neurons located in the gut. It's a highly densely innervated tissue. And you see both intrinsic neurons that I was discussing in the first part, neurons that are located inside the intestine. As you always see the processes coming out of the intestine, which represent the extrinsic innervation that are both feeding and coming from the intestine to the CNS and other sides. And the general distribution of these neurons, you can see here on the left side. As I was mentioning, the intrinsic enteric associated neurons located the cell body within the intestine. And then you have the extrinsic enteric associated neurons, which are neurons that are projecting to the intestine. And as an output, for example, here you have the sympathetic ganglia. It's called celiac ganglia or superior mesenteric ganglia, which is providing norepinephrine and TH innervation to the intestine. And you have, as well as you have the sensory arm of this enteric associated neurons, which is represented both by the DRG, the dorsal root ganglia, as well as the nodose ganglia. These are the two main hubs, sensory hubs, that can sense luminal stimulation in a variety of ways. And these two hubs are then connected to the CNS and regions of the brain, which I'm going to discuss at the end of the talk. So what Paul asked here is how come, and I briefly mentioned this question before, this sympathetic innervation, again, located outside the intestine, can really be influenced by the luminal content, particularly the microbial content of the intestinal tissue. And what we did, and we compared every extrinsic ganglia associated to the gut, is to first compare the gene expression levels in germ-free mice, animals that lack microbiota, with animals that are regular SPF mice. And in fact, when we compared several ganglia that are associated with the intestine, the sympathetic ganglia was the one that showed most differences between germ-free and SPF. These again are neuronal specific trap approach that I described in the first part of the talk. So here we are looking at active translating mRNA from neurons located in the sympathetic ganglia we believe that is innervating the intestinal tissue. And the surprising came when we saw the differences when you can see here some of the examples here, is that in the absence of microbes, we saw upregulation of neuronal markers that are associated with activation, particularly CFOS and ARC, which are markers of early neuronal activation. And that suggests that, contrary to what we would expect, absence of microbes would trigger the activation of these neurons. In fact, in data that I don't have time to present here, if you look at the intrinsic neurons located in the intestine, germ-free mice show less activation of intrinsic neurons. So this was counterintuitive to our expectations. So the first thing we did was to confirm whether you have indeed an activation of sympathetic neurons in the absence of gut microbiota. And the important point here, just to remind people that are not used to the relevance of the sympathetic branch, and it's known to regulate not only the gut but elsewhere several physiological processes, and I'm listing some of them here, including blood flow, motility, fluid balance, immunity, of course, many cells expressing adrenergic receptors, and also is known to regulate glia activation. So it's a neuronal circuit that is known to evolve and to regulate several physiological and likely pathological processes. So the first thing we did was to confirm that that was the case. In other words, that the microbiota could suppress the activation of sympathetic ganglion. And you can see here that germ-free mice isolated from different facilities. This we are comparing Jeremiah Fate facility in Mount Sinai with Rockefeller facility, very similar number of enteric neurons expressing CFOS, again, a marker of activation. And SPF mice coming from different facilities or maintaining different diets showed basically zero activation in steady state of these sympathetic neurons. This can be tunable by administration of antibiotics. So if you treat strains of mice, BOB-C, Black 6, with streptomycin and look the day after, you see already the release of the activation. They become activated, these neurons, which are not activated in a steady state. And if you leave these animals in the last bar here, 5 days without antibiotics, when you have the reconstitution of the microbiota, then is already sufficient to suppress again the activation of this, suggesting again that microbiota leads to suppression of activation of the sympathetic neurons that are innervated in the gut. The question Paul asked is, are these sympathetic neurons that are becoming activated exactly the ones that are projecting and sending norepinephrine fibers towards the intestine? And to address this question, what Paul did was a combination of a gene reporter and retrograde tracing experiment. So he injected chlorotoxin B, CTB, in the wall of the intestine, and chlorotoxin B would travel back from the fibers to the cell body. So that's a retrograde tracing experiment. And what we did was to combine that with antibiotic administration. Therefore, we could address whether the neurons that are connected, physically connected to the intestine, were also the ones expressing markers of activation. And what we saw, as you can see here, is that you see a lot of red cells, which are the ones that are connected to the intestine. This is in the sympathetic ganglia. Therefore, the fiber, the CTB travel from the fiber located in the intestine to the cell body located in the sympathetic ganglia in the control mice. However, in antibiotic-treated mice, you see a lot of green cells expressing FOS GFP. So these are the activated neurons, and you see by the green and red co-staining that most of these neurons are indeed connected to the intestine because they express the CTB. Of course, we have also green cells that are not expressing CTB, but that's because we are injecting the CTB in a small segment of the gut wall. So you would imagine that if you inject the CTB throughout the intestine, most of these neurons are likely to be the ones that are activated. So that shows evidence that not only the microbiota is regulating the activation of sympathetic neurons, but that also that these sympathetic neurons are exactly the ones that are sending norepinephrine fibers or TH fibers towards the intestine. So, of course, the question next is whether you need a diverse microbiota for this type of suppression. And we did several experiments to address that, and I'm going to show the summary of that. The first is that if you colonize, do a fecal microbiota transfer to germ-free with a regular bacterial composition from SPF mice, you can readily suppress the activation of sympathetic neurons. And if you compare two mice, strains of types of mice we had in the lab. One that had monocolonized with segmented filamentous bacteria. I guess most immunologists are familiar with this Th17 inducing bacteria. So single colonization with SFB was not sufficient to downregulate the sympathetic neurons. However, if you colonize mice with a consortium composed of 8 different bacteria that called altered shader flora, which includes a diverse set of microbiota species, that was sufficient to suppress sympathetic activation. And finally, we looked at, with the help of Greg Sonnenberg and David Artis in Cornell, at germ-free facility from them. And of course, as we observed in our facilities, there's a basal activation in germ-free mice of the sympathetic neurons. And when we looked at mice that were colonized with a Clostridium species consortium that was made by Kenya Honda in Japan, which is, by the way, it's known to induce regulatory T cell in the gut. So this Clostridium consortium was sufficient to suppress the activation of this sympathetic neuron. So what we conclude, and again, myself, I was skeptical about that because in the gut, most of these mediators are somehow ascribed to short-chain fatty acids. We conclude that the short-chain fatty acids are very likely a candidate that could be at least one of the mediators in the suppression of sympathetic neurons. Because all these combinations match what is known to produce short-chain fatty acids like altered shader flora and Clostridium, and also not produce short-chain fatty acids like SFB. So we further investigated this possibility, and what we did was to treat animals with single antibiotics used in an antibiotic cocktail. And you can see the names here: metronidazole, neomycin, ampicillin, vancomycin, and measure the amount of short-chain fatty acids with the help of the Memorial Sloan Kettering metabolomics facility of the short-chain fatty acid located in the cecum. And what we saw that ampicillin and vancomycin were the only antibiotics given individually that were able to really suppress entirely the production of all 3 main short-chain fatty acids located in the cecum. And you can see that also those 2 antibiotics were the ones that we observed that upon administration to animals would trigger the activation of the sympathetic neurons, again suggesting that these metabolites were the ones involved in the active suppression of sympathetic neurons. And to investigate this possibility with a different approach, I'm showing the 2 main ones here, which is to provide in the drinking water tributyltin, which is a stable form of butyrate that upon ingestion, this would release 3 molecules of butyrate in the gut. That was a better form of administration than in the drinking water. And what we saw both in animals treated with streptomycin that shows activation or germ-free mice at steady state show activation is that butyrate was sufficient, again, in the absence of microbiota, was sufficient to suppress the activation of gut-projecting sympathetic neurons. Okay, so the final part of this, and these are the final experiments for this presentation. It's a very neuronal-specific question that Paul, which is really a trained and really amazing neuroscientist had to address, which is how come that this, what are the secrets, potential secrets that this microbial or microbial metabolites can be using to suppress the activation of sympathetic neurons located outside the gut. And we actually investigated 3 possibilities. I'm going to show only the ones that gave the positive results. The first possibility that we looked and we did not find evidence for it is that sympathetic neurons would be directly sensing stuff happening in intestine or metabolites. We did not find evidence that this would be a sensory effect. Also, it's not known to be sensory neurons, the sympathetic neurons. They are mostly providing output of norepinephrine to the gut. We also look at the possibility that gut neurons that are projected to outside the intestine, these are called visceral fugal neurons, would be activating the neurons located in the sympathetic ganglia. We also did not find evidence for that. What we found to be the case is that sensory neurons that will be sensing either directly metabolites or molecules that were released, for example, by epithelial cells, with connected to regions of the brain that are known to be regions of a presympathetic control that are upstream of sympathetic chain. that upon activation would then activate sympathetic neurons. So that's a long loop from gut to sensory neurons to the brain, presympathetic control downstream from preganglionic neurons that would then activate this ganglia here. And I'm going to show a few examples of how we found or how we confirmed that this is the case. The first one, we used a DREAD animal, and the DREAD tool is used by neuroscience to modulate activation of neurons, and it can either activate or suppress the firing of neurons by providing a synthetic GPCR ligand. In this case, GPR is a compound 21, and you can do it in a cell-specific manner. We used many different CRE-mice, and I'm showing here the two combinations of CRE-mice, SNS-CRE and FOX2B, which are, according to our literature and also to our fate mapping experiments, are in combination, are preferentially targeting the nodose sensory neurons and avoiding other possibilities of sensing, for example, that DRG neurons would be sensing what is happening in the gut. So what we did was to use this DREAD tool to suppress the activation of these neurons. And in both cases, by suppressing the sensory arm of this circuit, again, we expect if you suppress the sensory arm, you mimic the absence of microbiota. Indeed, we see activation of sympathetic neurons in both cases here, as you can see by the measure of CFOS in the sympathetic ganglia. So that suggests that indeed there's an afferent sensory arm that is primarily located in the nodose ganglia that is required for the microbiota-mediated suppression of sympathetic neurons. Of course, if you pay attention to the first part of the presentation, I mentioned that sympathetic neurons, upon release of norepinephrine, can be modulating this macrophage activity, which is neuroprotective. So we, after discovering that this is a possible way of modulating sympathetic neurons, we asked whether, coming back to the first part of the presentation, whether the activation of sympathetic neurons by this DREAD tool could work in preventing neuronal cell loss. And that's a parenthesis in this part of the presentation, but just to show that we indeed observe that. So if you activate gut-projecting sympathetic neurons with these DREAD tools using SNS or FOX2B CRE-mice and infecting animals with SPI-B, then we did not observe neuronal loss upon infection. Again, demonstrating one of the potential functional consequences of modulating sympathetic neurons in the gut. So the final question are, what were the neurons that were upstream of the sympathetic control? In other words, what were the neurons that the gut sensory nodose ganglion would be projecting the CNS that would regulate the sympathetic ganglion? What Paul used to answer this question was a neuroscience classical tool of pseudorabies virus, which you can use a multisynaptic tracing to really investigate what neurons that are indirectly connected to the gut that are located in the brain. So what you do is you inject this pseudorabies virus expressing RFP in the gut wall. Within days, you see the first order connections being light up, and that will include sympathetic ganglia, dorsal root ganglia, nodose ganglia, which are connected by one synapse to the intestine. And then if you wait a few more days, you see which neurons in the gut are connected with that. Which neurons in the brain are indirectly connected to the intestine. So we use a clear view tool, and this is an eye disc of the entire brain of these animals. And you can see here, these are 4 days after PRV injection in the gut wall, we start seeing neurons being highlighted, and that will identify a region of the brain that would be indirectly connected to the gut. And again, I'm sorry for the number of jargons in neuroscience I'm using here, but we basically identified a few regions in the brainstem that could be relevant as a potential upstream of the sympathetic control. And 2 sets of regions, the raphe pallidus in the center here, and this nuclei here called gigantocellular nuclei, were preferentially expressing the inhibitory, as you can see here by the GFP or the VGAT, inhibitory GABAergic neurons in the brainstem, and they also co-express the PRV. And the second main nuclei that we observed that was labeled by the PRV was this lateral more distal from the raphe, lateral gigantocellular nuclei, which were preferentially expressing excitatory neurons expressing VGLUT2, as you see here by the use of the VGLUT2 reporter mice. So there are two contrasting neuronal populations within the brainstem that were indirectly connected to the gut and potentially upstream of the sympathetic control. And we addressed whether these two populations, which one of the two populations could be mediating the regulation of gut projecting sympathetic neurons. And what we did was to inject another dread tool, in this case is an excitatory dread. So this dread HM3 would induce activation upon administration of this synthetic GPCR ligand. We injected in the VGAT3 in the nuclei gigantocellular, which is the proximal nuclei, because that's where we saw the correction of the PRV. Or we injected this excitatory dread virus here directly in the region that we saw expressed in the VGLUT2 Cre mice. So therefore, in this case, we would be able to activate specific neuronal populations that we saw to be connected to the intestine and test which of these two populations were regulating the sympathetic neurons. Again, just to point out that this dread tool here is exactly the opposite of what I showed before, which was inhibitory dread. This is an excitatory dread. So you provide the ligand and you trigger firing, neuronal firing. And what we saw, and again, to make a long story short, is that the inhibitory GABAergic neurons upon activation did not lead to any change in the parameters that we investigated. However, the activation of these excitatory neurons located in the lateral gigantocellular nuclei of the brainstem would trigger both change in motility, and the release of norepinephrine is known to delay motility, and that's what we observed, as well as activation of this gut projecting sympathetic neurons. I know that's already 1 p.m. Well, in the East Coast, and that's the final experiment just to prove that we have a loop. What Paul did was to inject fiber-filling virus AAV9 in the nodose ganglia and see whether this nodose ganglia would be projecting to regions where you have rich short-chain fatty acid concentration, like in the proximal colon. That's what Paul saw. That the fibers coming from the right, particularly the right nodose ganglia, that is also from the left nodose ganglia, projecting to the proximal colon. You can see here the image of that. So you inject this virus in the sensory nodose ganglia, and the protein would travel all the way to the colon, suggesting that there are sensing fibers really very close to the main stimulation, which is short-chain fatty acids in this case. And finally, these are regions of the brainstem that are known to be connected to the nodose ganglia, the adipostrema and the nucleotriatus solitarius. And both regions are activated when you treat mice with streptomycin. Again, suggesting that the nodose ganglia not only sensing what is happening in the gut, but also projecting to the regions of the brainstem that are then connected to this nuclei that you characterized in the slide before. So the final conclusion of this, and I'm sorry for very heavy neuroscience second part here, is that in the presence of microbiota, short-chain fatty acids, there is engagement of a circuit that involves nodose ganglia that connects to the regions of the brain that are constantly suppressing the activation of sympathetic neurons. In the absence of the microbiota, this circuit is it's disrupted, and you have release of gut projecting sympathetic neurons activation, which then, of course, will induce norepinephrine secretion and delay motility in the gut. So I will stop here. I already thank most people directly involved with the project. There are people that also should be mentioned in the lab. For example, Thiago, that helped with the bioinformatic analysis, and several other people that are in this picture that helped in different ways. And really, it's a pleasure to have such diverse lab and hardworking people that are in different parts of the world. In fact, Tomasz, for example, is stuck in Poland because of the travel ban. Anyway, everybody at least healthy. So let's see when we're gonna end this this crisis. And these are the people that I mentioned before that help the lab with several different tools, and I'm really thankful for all of them. And I also would like to mention Michelle, Juan, and Gabriel, where we have people that we have joint lab meetings, particularly Juan and Gabriel, with all the discussion and help with really developing this project. The final thank would be with really for Jeff Friedman's lab, who really in half of the Jeff's lab has engaged in this project, and particularly Mark Payne, who really is involved in several aspects of this specific injections in the brain and modulation of neuronal activity. So really thankful for Jeff Friedman's lab. And I guess there is no questions in this presentation directly, but the questions are, I think, are being asked in the Twitter. My Twitter is Dan Mucida, and I would be happy to answer these questions as I stop here and have some lunch.
Perfect. Danny, this was really a spectacular talk. I definitely learned a lot. I'm sure you will have tons of questions from the attendees, so that will go. Here is the hashtag Global-immune and Danny's Twitter account. So please go ahead with the questions. And Danny, thank you so much again. It was spectacular. Thank you. Really appreciate. And I hope to see everyone next Wednesday again. Same time. Right? Say again.
Caetano is presenting next week.
Caetano Reyes Souza is presenting, so another great speaker. And so this, we are looking forward to that, and we hope to see everyone again. And please help us spread the word with this Global IMMUNOTALK so more and more people can benefit.
Yes,
okay, so with that, thank you so much. So we'll close.
Thank you, Danny.
Yes.
Bye, everyone. Thank you.
Bye-bye.